Okay, part 2 is Hardy-Weinberg equilibrium. So in general, allele frequency and genotype frequency are independent from each other. So if I gave you a population like I did before, if I gave you a population of frogs and I asked you to calculate the genotype and allele frequency from that, you should be able to do that. And so you do this just kind of taking the approach that I showed you. So you count the number of animals with each genotype and then you divide by the total number of animals. And then from this, you can calculate the allele frequency from that. And so what do I mean by the fact that these are independent? So what I mean by this might be easiest just to illustrate by an example. But let's say we had a population that had 32 individuals. And so this is the number of individuals, and then so I'll just do I for individuals, and then the number of alleles I'll just do as A, and so that would be 64. And so let's say I told you that 2 big A alleles were in the population and 62 little a alleles were in the population. And then I wanted to take that number and I wanted to say, okay, what is the genotype frequencies? And so you can't really answer that question because there's multiple possibilities. So in this case, one possibility is this. Oh, sorry. I'm not sure. Okay. And one possibility is this. All right, so in the first case, one individual is homozygous dominant and 31 individuals are homozygous recessive. And then in this other case, 2 individuals are heterozygote and 30 individuals are homozygous recessive. And so this is what I mean. In both cases, if we calculate the allele frequency, there's 2 alleles of the big A and there's 62 alleles of the little a. But you can see that the genotype frequencies are different from each other. And if we expand this, if we make it something like 4 and 60, then there's even more. So for this one, there's 2 different possibilities. But if we have 4 different big A alleles and 60 little a, then there's even more genotype possibilities from that. And so this is what I mean by they're independent. This is the concept I'm trying to get you all to understand, is that an allele frequency can have multiple different genotype frequencies, and there's multiple genotype frequencies that can result in the same allele frequency. So Hardy-Weinberg equilibrium is a special solution where the allele frequency determines the genotype frequency. So in this case, if I tell you that a population is in Hardy-Weinberg equilibrium, then all I have to give you is the p value, and then you should be able to figure out everything else. So let's go over this. So let's say I told you p equals 0.2 for A. And now I said figure everything else out. Figure out all of the genotype frequencies and figure out all of the allele frequencies. So the first thing that you should do is you should calculate Q, and the way you calculate Q is you just rely on the fact that these sum up to 1. So 0.2 plus 0.8 equals 1. So I now know that 20% of the alleles are big A and 80% are little a. And then to calculate the genotype frequencies, I simply use this relationship right here. So the frequency of big A is p squared. The frequency of big A, little a is 2pq. And the frequency of the recessive homozygous is q squared. And so this is essentially just using a Punnett square in order to calculate this. So before we said the size of the Punnett square are the possible gametes. So there's 2 possible gametes, big A and little a. And so then the frequency of each would just be P or Q. So the probability of just randomly picking out a big A is the frequency in the population. So it'd be 0.2 for this particular case. And then the same thing is true on this side. And so if you just group these together, p squared, and remember just multiply these together, p squared is in front of the big A, PQ, and then since there's two that have the same genotype, the heterozygote, then it's 2 times P times Q. And then for the Q squared, it would just be, or sorry, for the little a, it would just be Q squared. And so, you know, just reminder, the allele frequencies add up to 1, and then the genotype frequencies also add up to 1. And so for a population to be in Hardy-Weinberg equilibrium, it has to be infinitely large. So these are not real populations, obviously. There's no infinitely large populations. But what we're trying to do is we're trying to ignore the effects of stochasticity. So we know if we have a finite number of individuals and we just randomly sample something, then you know there's going to be some sort of noise. But if we have an infinite large populations, then we don't have to worry about that. The other thing that we have to worry about is random mating. And so random mating means that individuals, when you pair up for mating, they just randomly choose another mate. There's no sort of sense within that. And so what that means is that AA mates with this genotype, and this genotype, kind of without regard. Okay, so, you know, there are certain types of mating, associative mating, for example, where individuals might choose to mate with individuals given their phenotype. So an example of this is, you know, think about pea plants. And so, you know, tall, and then short. So associative mating would mean that maybe talls only like to mate with tall individuals and short individuals like to mate with short individuals. So that is possible, but though that population would not be in Hardy-Weinberg. So what Hardy-Weinberg means is that these tall individuals don't care what the phenotype of the mate is. They just randomly choose it just in a completely random process. And so the frequency of these matings would be determined by the genotype frequencies in the population. And then the final thing that you have to worry about are that there's no evolutionary forces. So there's no genetic drift, there's no migration, there's no selection, natural selection going on, because all of those things can also influence allele frequencies. And so those can also skew them away from these ratios. But if all these three things occur, if all these three conditions occur, then now we can predict with certainty what the genotype frequency is using allele frequency. And very often, as you'll see later, we assume that this is the case. So we're going to assume that populations are in Hardy-Weinberg equilibrium in order to make certain predictions. So let's go over a couple examples. So one example of using Hardy-Weinberg is resistance is trying to predict resistance to HIV-1. So human geneticists have found a gene CCR5, and this gene, which is encoded by multiple exons, it's a human gene, it has an allele called the delta 32 allele. So CCR5 encodes a receptor for strains of HIV-1, so the way that viruses often find the cell that they infect and are able to actually gain entry into a cell to hijack its process is that it physically binds to some sort of transmembrane receptor that is present on the outside of the cell. And so for HIV, they recognize this protein CCR5. And so individuals that are homozygous for the delta 32 allele are resistant to HIV infection. Heterozygotes are susceptible to infection but progress more slowly to AIDS. And so this is the deletion of about either 32 base pairs or 32 amino acids. And this deletion prevents the HIV from binding to it. So you can actually determine the genotype of an individual just by using PCR and restriction enzyme digest analysis. So you can PCR up a portion of the CCR5 gene that contains this deletion, and then this deletion contains a restriction enzyme site. So individuals that don't have the deletion, you'll be able to cut the DNA here, but if they have the deletion, that restriction enzyme site is gone. And so it's a very straightforward, something that's very straightforward to genotype. And I'm not sure if you followed the CRISPR-Cas9 controversy where human babies were engineered and were actually genomically edited, and the choice of this edit was this delta 32 allele in order to, because they were born from HIV positive parents, and so it was one way to ensure that the parents did not, in case the parents did transmit HIV to the offspring, those offspring should have been resistant to HIV infection. So, here is an example of the genotyping that you can do. So 1-1, that's the wild type allele. So these individuals are susceptible to most strains of HIV-1. These individuals are homozygous, and so they are resistant to most strains of HIV. And then these are the heterozygotes that are susceptible but progress to AIDS more slowly. So it confers some level of resistance to HIV. So, and then in this case, there's not actually a restriction enzyme site. So this is simply using the size of the band as a way to infer. Sorry, this is a restriction enzyme site. I apologize. So this is the way this gel was produced, is that you had a large piece that was PCR'd up. So a 735 base pair piece of DNA that was PCR'd up. And then if the individual had the deletion, what was PCR'd up instead was a 703 base pair piece of DNA. So this deletion, this 32 base pair deletion, when you PCR amplify up a portion of that gene that contains the deletion, you have two different size fragments. And so actually I think I gave you a little bit misleading information in the previous slide. This restriction digest site is not actually in the deletion, but it cuts the piece so that it's easier to resolve on a gel. And so it cuts these large pieces into 332 base pair chunks. So one size is 332 base pairs, and then the other side is either 403 base pairs or 371 base pairs. And so this is how this gel arises, is that in both cases you have this chunk right here, the 332 base pair piece, but depending on whether you have the deletion or not, you either have a 403 base pair band or a 371 base pair band. So you can see the homozygotes that are wild type, the homozygotes of the deletion, and then the heterozygotes, because we're diploid and we have copies of both, you end up seeing both of these bands. And so this is just a simple way of using gels in order to genotype this. All right, so long explanation. And so now what are we going to try to do with this information? So what are the genotype frequencies and the allele frequencies within a population? So here is an example of 100 individuals that were genotyped for this allele. And so you can see of the 100, only one of them was homozygous recessive. So this is actually a very rare allele in the population. Only 1 out of 100 individuals actually were homozygous recessive. So only 1 out of 100 individuals would have been resistant to HIV. And so this is something you obviously can't rely on. So if you are an individual and you get HIV, most likely you're not going to have that protective genotype. 20 of the individuals are heterozygote, and then 79 individuals are homozygous for the wild type, the normal wild type allele. And so we can use this to calculate the genotype frequency, and we can use this information to calculate the allele frequency. And so if we want to calculate the one allele, how many one alleles there are, again, we just multiply 79 times 2, because each of these individuals has 2. Each of these individuals has 1 of this, so you have 20 right here. And then these individuals have 0, because they're homozygous for the other allele. And so you simply add up 158 plus 20 plus 0, and you get 178. And then you can do that same procedure for the delta 32. So there's 0 alleles from these, 20 alleles from these, and then 2 alleles from this single individual, and they add up to 22. So to calculate the allele frequency, you just take the 178 and you divide by 200. So P equals this, and then Q equals this. Equals this. Okay. So 22 divided by 200 is 11%, and 178 divided by 200 is 89%. So P equals 89%, and Q is equal to 11%. All right, so we have calculated the allele frequency, and then if you wanted to calculate the genotype frequency, you would just divide these numbers by 100, since there's 100 individuals. So 79% are this genotype, the 1-1, 20% are heterozygotes, and then 1% are the delta 32 homozygotes. So what are the expected genotype frequencies if we used Hardy-Weinberg? So just as a reminder, this data that we just calculated, this was using actual data. So we'd never used Hardy-Weinberg to calculate either the genotype frequencies or the allele frequencies. But now we can ask the question, what are the expected genotype frequencies if we use Hardy-Weinberg equilibrium? And so we're going to take this data, we're going to rely on this allele frequency data, 89% and 11%, as our P's and our Q's. And so we're going to calculate the genotype frequency, the expected genotype frequency, by just using P squared, 2PQ, and Q squared. And so we can calculate this. So 0.89 times 0.89 is equal to this. The expected genotype frequency for the heterozygote is this, and the expected homozygote recessive, so the homozygote delta allele is this. And so we can compare these to the actual values. So this was 79%, 20%, and 1%. So you can see for this population, they actually fit very close to the expectations from Hardy-Weinberg. And so one of the consequences of this is that even if allele is somewhat present, so 10% of the alleles are this allele, it's very rare to have an individual that's homozygous for it, because you're squaring this number. So this 11% goes down to 0.01% because of that. You can also handle 3 alleles for Hardy-Weinberg. And so how do you do that? So what is a situation where you have 3 alleles? So you have A1, A2, and A3. So we're no longer in the biallelic, where there's just a big A and a little a. We're now in the triallelic, where there's A1, A2, and A3. So we just add a letter. So P, Q, and R are now the allele frequencies for those 3. And so the new equation becomes P plus Q plus R is equal to 1. So no longer is P plus Q equal to 1, because you have to have this third allele to worry about. But if you add up all the allele frequencies together, you get 1. And so if you wanted to calculate the predicted genotype frequencies, again, it's just you use the Punnett square, just kind of how we talked about, back in chapter 4 and chapter 5. So you have 2 sides. There's now 3 different gametes that can be produced. So you can either be A1, A2, and A3. And the expected probability of producing that gamete is just P, Q, and R. and then the other individual would have the exact same frequencies. And so now all we do is we just fill out this square, the inside of the square, so p times p equals p squared, and this would result in an A1 homozygote. And we fill this out for the entire thing. So if we do this, we can calculate the predicted frequencies for each of these. So A1 is p squared, A1 is p 2pq, A1 for example, is 2pr, and so on and so forth. So I can let you do the rest of that exercise, just by grouping these. But you can see all I did to calculate A1 and A3 is I just grouped these two together and then added together to get 2pr. For the A1, A2, similarly, I just grouped them together to get 2pq. All right, so how can we use that? So an example is blood type. So blood type is an example of a 3-allele locus. So just as a reminder, there's the IA, which confers the A blood type, IB, which confers the B blood type, and then the little I recessive. So A and B are codominant to each other. So if you have both alleles, your blood type is AB. Little I is recessive, so only if you're homozygous recessive do you have the O blood type. So the blood types for these, just as a reminder, are given as so. And so we can use Hardy-Weinberg to predict the genotype frequencies if we were given the allele frequency. So this data right here is where we're given the allele frequencies. And so just as a reminder, this is P, this is Q, and this is R. And so the IA, that's P squared. And so 0.14 is how many individuals we expect to be homozygous. The heterozygote is 2PR. And so these are the number of individuals that we expect to be heterozygote. And so if we wanted to calculate how many individuals we expect to have the A blood type, it's 53% just by adding these two numbers together. Similarly, to get the B blood type, we simply add these two numbers together. So again, we simply just use the allele frequencies to calculate the expected genotype frequencies and then group these together because these two genotypes have the same phenotype, which is the B blood type. And then the AB would be this, and then the O would be this. So Hardy-Weinberg allows us to take the allele frequencies and then turn that into genotype frequencies, which allowed us to predict the frequency of blood types in a population. So again, this is only possible if a population is in Hardy-Weinberg or you're given the express instructions, assume that the population is in Hardy-Weinberg. So if I don't tell you that it's in Hardy-Weinberg, you typically have to actually calculate the genotype frequencies using data from individuals. All right, one final example of how we can use Hardy-Weinberg in human populations. So cystic fibrosis, so this is an autosomal recessive disease. And so the incidence, so cystic fibrosis we know is autosomal recessive, so we've analyzed all of these pedigrees, and by analysis of the pedigrees, those were all consistent with an autosomal recessive disease like we discussed in previous chapters. And so if we go through the population and we calculate how many individuals actually have it, it's 1 out of 2,600 people from a population like a Northern European population. So it's a very low percent, so it's 0.0004 people will have this disease. And so we might want to know, so we have this data, this is our data, and what we might want to know is what is the number of carriers that are in the population. So if I told you 1 in 2,600 people have the disease, then I might ask you the question, how many people are carriers for the disease? And so we can use Hardy-Weinberg as a way to estimate that. So again, this is just an estimation because the human populations are not expected to be in perfect Hardy-Weinberg equilibrium, but we can estimate that if they were, then this is what the allele frequencies would be. And so the genotype frequencies are just given by these three numbers. And I've told you that the CC, so the number of individuals that have the disease, is 1 out of 2,600. And so that means that Q squared is equal to 1 over 2,600. And so now we can solve for Q, and then we can solve for P by just knowing that 1 plus Q is equal to, P plus Q is equal to 1. And so the number of carriers, so these are the allele frequencies, and so the number of carriers is given by 2pq. So the number of heterozygotes are the carriers, and so now we just do 2 times this, and so it's approximately 4%. Not quite 4%, but pretty close to 4%. So 4% of the population, so if we were all in a room together, a large non-zero number of individuals within this room would actually be a carrier for cystic fibrosis.